Part 1, Microbial Growth and Metabolic Processes. Microbes grow and thrive in many different environments. Two major types are anaerobic bacteria. These can't live in oxygen-rich environments. You might find them in canned foods where there's no air. Facultative anaerobes. These bacteria are flexible. They can live with or without oxygen, like the ones in raw meat. Microbes use different metabolic pathways to break down nutrients. Some use anaerobic respiration, while others ferment sugars. Scientists use metabolic tests, like color-changing media, to identify which microbe is present. It's like using your nose to sniff spoiled milk. You can tell a lot by what's being produced. Mini review. Anaerobes equals no oxygen. Facultative microbes, flexible with or without oxygen. Metabolic tests use color changes to show what the microbe is doing. Quiz Part 1, Growth and Metabolism. Q1, where might you find anaerobic bacteria? In canned food. Q2, what kind of bacteria can survive with or without oxygen? Facultative anaerobes. Q3, what does a metabolic test do? It shows how bacteria break down nutrients using color changes in test media. What's a real-life example of using senses to identify bacterial activity? Smelling spoiled milk. Part 2, Microbial Genetics Overview. Let's talk about DNA. Bacterial DNA is circular, not straight like in human cells. Found in a region called the nucleoid in the cytoplasm. Is supercoiled, meaning it's tightly twisted to fit in the cell. DNA is made of 4 nucleotides: adenine A, thymine T, cytosine C, guanine G. They pair up, A with T and C with G, like puzzle pieces. Hydrogen bonds hold them together, while covalent bonds make the DNA backbone strong and stable. Mini review. Bacteria have circular supercoiled DNA. It's located in the nucleoid. A pairs with T, C pairs with G. Quiz Part 2, DNA Basics. What shape is bacterial DNA? Circular. Q2, where is bacterial DNA found? In the nucleoid region. Q3, what nucleotide pairs with adenine? Thymine. Q4, what type of bond holds base pairs together? Hydrogen bonds. What bond makes the DNA backbone strong? Covalent bonds. Part 3, DNA replication and genes. DNA replication starts at a point called the origin, ORI, and ends at the terminus, ter. The enzyme DNA polymerase does the copying. It works fast, around 600 nucleotides per second. It's accurate, reducing mutations. Each bacterial genome has about 4 million base pairs and around 2,000 genes. Genes are sections of DNA that contain instructions to make proteins, which run all cell functions. Mini review. DNA replication starts at ORI and ends at ter. DNA polymerase is the key enzyme. Bacteria have millions of base pairs and thousands of genes. Quiz Part 3, DNA replication. Q1, what is the enzyme that copies bacterial DNA? DNA polymerase. Q2, where does replication start and stop? Origin, ORI, and terminus, ter. Q3, how many genes are in a typical bacterial chromosome? A3, about 2,000 genes. Q, what do genes do? They code for proteins. Part 4, evolution and genetic diversity. Bacteria reproduce asexually. They clone themselves. That sounds limiting, but they still evolve quickly through mutations, errors during DNA copying, horizontal gene transfer, DNA sharing between bacteria. In sexual reproduction, like in humans or plants, genes mix every generation. This helps create genetic diversity. Diversity matters. Take the potato blight. A genetically identical potato population was wiped out because it lacked resistance. Bacteria solve this by exchanging DNA constantly. Mini review. Bacteria reproduce asexually. Genetic diversity comes from mutations and DNA sharing. Sexual reproduction increases variation. Quiz Part 4. Evolution. How do bacteria reproduce? Asexually. What are 2 ways bacteria create genetic diversity? Mutations and horizontal gene transfer. Why is genetic diversity important? It helps populations resist diseases and adapt. What historical example shows the danger of low genetic diversity? The Irish potato blight. Part 5. Antibiotic resistance. Bacteria evolve resistance to antibiotics like MRSA, resistant to methicillin, VRE, resistant to vancomycin, Pseudomonas strains. These changes come from mutations and gene sharing. The problem? It makes treating infections harder. Hospitals constantly face bugs that are resistant to multiple drugs. Mini review. Bacteria can evolve resistance to powerful antibiotics. Resistance spreads through mutations and DNA exchange. Quiz Part 5, Resistance. Q1, what does MRSA stand for? Methicillin-resistant Staphylococcus aureus. How do bacteria become resistant? Through mutations and gene sharing. Q3, why is resistance dangerous? It makes infections harder to treat. Part 6, Bacterial gene exchange. Bacteria have 4 key methods of sharing DNA. 1. Plasmids, small, circular DNA fragments, carry genes like antibiotic resistance, can survive after the bacteria dies. NAR2, F plasmids in conjugation, larger plasmids with special instructions, transferred via an F pilus, like a tiny tube between bacteria. 3. Viral transduction. Bacteriophages inject their DNA into a bacterial host. This DNA may carry toxins or resistance genes. 2 viral paths. Lytic, virus kills the cell. Lysogenic, virus DNA hides inside host DNA. 4. Transposons, jumping genes, pieces of DNA that move within or between genomes, found in bacteria, corn, even humans, cause random mutations and diversity. 5. Recombination, 2 DNA strands get tangled and exchange parts. Not as common, but adds to variation. Mini review. Plasmids, conjugation, viral transduction, transposons, and recombination all help bacteria swap DNA. Quiz, Part 6, Gene exchange. Q1, what are plasmids? Small circular DNA pieces separate from the bacterial chromosome. Q2, how are F plasmids transferred? A2 and F pilus in a process called conjugation. Q3, what is viral transduction? Bacteriophages inject their DNA into bacteria. What's the difference between the lytic and lysogenic cycles? Lytic kills the host, lysogenic hides in the host DNA. Q5, what do transposons do? Jump around the genome, causing random mutations. What is recombination? Exchange of DNA segments between 2 strands. Final recap. You just covered bacterial metabolism, DNA structure and replication, genetic diversity through mutations and exchange, how resistance evolves, 5 major mechanisms of gene exchange, plasmids, F pilus and conjugation, viral transduction, transposons, recombination, and most importantly, bacteria are constantly evolving. Let me know if you want